First of all, I want to thank the organizers for the opportunity to speak here today. And yesterday, Volker Krömker told us something about persistence and reinfection. And referring to this, I would like to present our study about Streptococcus uberis and its subtypes, which was conducted in Austria. So, what do we know about Strep uberis? It's one of the most important mastitis pathogens worldwide. It causes up to 25% of clinical and subclinical intramammary infections. And from practitioners' point of view, it causes several problems in therapy. Poor response to treatment is reported, and despite therapeutic attempts, recurrent cases of mastitis are frequently observed. This can be explained by treatment failures, of course, but we cannot exclude mastitis due to reinfection or due to persistent strains. To get a further insight in these aspects of Strep uberis mastitis, molecular analysis is needed. In epidemiological research, subtyping is a common way of analysis for differentiation beyond species level. This means you can characterize your Strep uberis strain more exactly. For example, this mastitis is caused by Strep uberis subtype number 2. There have been several typing methods used to characterize Strep uberis, for example, the so-called pulse field gel electrophoresis, or abbreviated PFG. With this technique, you cut the genome with specific enzymes and create something like a DNA fingerprint, but without a complete sequence analysis. PFGE is based on genetic relationships and is reported to have a high discriminatory power and is therefore very suitable for Strep uberis. This research was usually conducted in large dairy herds, for example, in New Zealand, Australia, the United States. And for that reason, Subtyping studies reveal two different observations. On the one hand, there's a great diversity in and among dairy farms described. Recurrent mastitis is explained with reinfection. And this supports the accepted convention that Strep uberis is an environmental pathogen and hygiene problems lead to an increase in mastitis cases. On the other hand, working groups found strains predominating on farms. Persistent infection could be observed, and Strep uberis spread within a herd by the milking machine has been discussed, suggesting that cow-to-cow transmission may occur. And this is a kind of paradigm shift that Strep uberis can show characteristics of cow-associated pathogens like Staph aureus. So now, this is the right slide, I'm sorry. This research was usually conducted in large dairy herds, for example, in New Zealand and the United States. And for that reason, we wanted to determine the Strep uberis variety in small herds that are typical for Austria and a large herd in comparison. Our hypothesis was that Strep uberis strains are a heterogeneous group, but some clones are predominant, host-adapted, and widespread. In our study, 311 isolates were included. They were obtained from routine screening of quarter mix samples, as well as herd visits. And 2/3 originated from our small farms with an average of 30 cows. And the other third was collected in a large Slovakian dairy operation close to the border, as you can see on the picture. They were collected from clinical mastitis cases. And their affected cows were treated with beta-lactams and resampled 2 and 3 weeks later for follow-up examination and checked for Strep uberis again. All these isolates were confirmed with 16S gene sequencing as Strep uberis with the gene bank database. Subtyping was conducted with a modified SMA-based protocol. SMA is the name for the enzyme for the macro restriction, and the pulsotypes were evaluated with cluster analysis. Such cluster analysis is shown on the top side of the slide. PFGE of 311 isolates revealed 204 different profiles. 187 were found in the small Austrian farms and 17 in the large dairy operation. And unfortunately, 8 had to be excluded because they could not be evaluated. 158 of these profiles appeared only once each, which is a high amount of heterogeneous Strep uberis profiles. They were found mainly in the small farms in Austria. And we found 38 subtypes highly abundant, which means they were repeatedly isolated from mastitis cases, both in small and in large dairy herds. At more detailed level, we detected different subtypes in different quarters of the same udder. We also found the same types both within and between farms. We named this intra- and inter-farm spread. These farms were not related in any way, for example, due to animal movements or mixed pasture. And mainly in the large herd, we found 10 types predominating. This means they could recurrently be isolated from the same quarter within the 3 study weeks on mastitis days 0, 14, and 21, and this despite treatment. Basically, all these major types were herd-specific. So, how do we explain these findings? The high amount of unique subtypes is possible due to self-limiting mastitis or to successful treatment, and these are very good news for the veterinary practitioners because the biggest proportion of Strep uberis strains seems to be curable. Interestingly, the major types were herd-specific, which indicates infection from the same reservoirs. This could be another cow or the environment. Regarding the different types in the same udder but different quarters, and the various types in different cows of one herd, there could be a link to an environmental reservoir, for example, straw bedding or feces as source of infection. And this has been shown in previous studies where Strep uberis was found almost everywhere in cows' environment, their gut or skin. These strains might cause intramammary infection due to contamination of the gland as a result of poor environmental hygiene conditions. In contrast to this, there were also subtypes which were detected for several sampling types. This finding indicates persistence or immediate reinfection with Strep uberis. Reinfection with the same subtypes from the environment is extremely unlikely with the high number of unique subtypes we found. However, we cannot exclude that a transmission may also occur during milking, but we prefer the theory of persistence in the quarter. Unfortunately, there's nothing available that can help us to distinguish which is true. Finally, to conclude, Strep uberis mastitis is caused by herd-specific subtypes, and there is persistence and contagious transmission may occur, but we cannot say why and what are the key features are Strep uberis strain needs to persist. Therefore, we would be happy if there were infection challenges or further analysis with these strains in future to answer these questions. Thank you for your attention. I've come to the end. Would be very pleased to receive comments or questions.